Microbes have an incredible ability to exchange DNA with other microbes throughout their life. Humans and other higher organisms, we can't do this. I cannot change my eye color by stealing DNA from my green-eyed lab mate. But as you can probably imagine, being able to acquire a completely new function throughout life is a powerful tool for surviving the many inhospitable and ever-changing environments that microbes inhabit. The ways in which microbes exchange DNA are numerous, but this project focuses on a really unique mechanism of what we call horizontal gene transfer. It's unique in that it allows microbes to exchange really large pieces of DNA between distantly related microbial species. But what's really fascinating here is that this process appears to be more prevalent in microbes that live in extreme environments where other organisms can't survive. So this begs the question, is there something about horizontal gene transfer or the ability to acquire DNA from your neighbors that allows microbes to colonize or invade environments that might otherwise be inaccessible? Meet Halopharax vulcanii. This pink microbe is an extremophile isolated from the Dead Sea. It loves growing at high temperatures and high salt concentrations. And of course, it readily acquires DNA from its fellow salt-loving neighbors. So we actually witnessed horizontal gene transfer for the first time 25 years before we discovered DNA. But historically, microbiologists thought that this process was extremely rare. However, improvements in genome sequencing over the last 20 years have allowed us to peer into microbial genomes at an unparalleled resolution. And one of the most surprising things that we found is that microbial genomes are actually complex mosaics of DNA likely acquired from many different microbial species. And this is really exciting because it calls into question the long-held notion that horizontal gene transfer is rare. I think it's a really exciting time to be an experimental biologist working on horizontal gene transfer, because on one hand, the evidence is right in front of us in the genetic code. But from an experimental standpoint, we really don't understand why microbes undergo this process or how often they undergo it, especially in multi-species communities. And that's where this experiment comes in. The experiment begins by creating mock communities of salt-loving microbes to which we introduce our model organism, Halopharax vulcanii. To half the communities, we introduce a strain capable of horizontal gene transfer, or mating, as we call it in the archaea world. To the other half, we introduce a strain that can't undergo mating. Over 2 months, we track the abundance of Halopharax vulcanii. We expect that by the end of the experiment, the strain capable of horizontal gene transfer, or mating, will establish at a higher abundance, meaning it's a better invader. In order to establish if horizontal gene transfer played a role in this, we can sequence the DNA of each Halopharax vulcanii strain and screen for candidate horizontal gene transfer events that may have improved invasion success. We have to keep in mind that even though horizontal gene transfer is a really interesting process, it also poses a significant risk to human health. Horizontal gene transfer is a major driver in the spread of antibiotic resistance genes, and many human pathogens undergo horizontal gene transfer at very high rates. Even evidence from the COVID-19 pandemic suggests at least 11 different horizontal gene transfer events contributed to the emergence of SARS-CoV-2. But horizontal gene transfer is not only important for human health, But studying horizontal gene transfer can also teach us about the fundamental processes that govern evolution. Specifically, high rates of horizontal gene transfer suggest that evolution only needs to find the solution to a problem one time. And once that solution is found, it can spread to many organisms through horizontal gene transfer. And that is something that's really powerful.